All right, so here I'm just going to talk about some other RNAs that are not typically thought about very much. We typically think, when we think RNA, messenger RNA, transfer RNA, ribosomal RNA, but there are some other RNAs that are very important. So we're going to talk about small nuclear RNA. There is also small nucleolar RNA and microRNA. We'll spend most of our time on microRNA. So small nuclear RNAs are going to be important in splicing out introns from eukaryote transcripts. And there are many of these nuclear RNAs that are going to be important, and they work together with our type 3 introns in that spliceosome. We also have small nucleolar RNA that are going to be encoded actually in some of the introns of our genes. And they are going to be involved in modification of your ribosomal RNA, your transfer RNA, and your small nuclear RNA. So these are going to be primarily found in the nucleolus, and so where you have processing of those RNA products. And then finally we have our microRNAs, and these are going to be involved in gene regulation. So microRNAs are going to be a non-coding RNA. They are going to be made at a very long nucleotide, but eventually they will be cleaved down to about 22 nucleotides long. These are made to be complementary to messenger RNAs, and they are going to be regulating the messages that they are complementary to. 1% of the genome is going to encode for these microRNAs, and they're thought to target about a third of all of our messenger RNAs transcribed. And so this is a very important regulation of our messages, which of course means that they're regulating proteins. Now microRNAs are going to be formed, and they're going to be encoded in our DNA, and then they are going to be formed into a pre-microRNA, and then they're going to be targeted to eventually the cytoplasm. Now, there are many enzymes involved in this, DROSHA, dicer, things like that, that if you want to get down to the molecular, you can. But in essence, what's really important for medical biochemistry is that you will get a mature microRNA complex. And why this is important is because the mature microRNA is going to have two results, depending on how closely it matches the messenger RNA it's trying to target. So if this mature microRNA is going to target nearly perfect to the messenger RNA, you get almost lining up perfectly, perfect complement, you're going to get degradation of your messenger RNA. It's going to simply be degraded. If it's partial, so you get these kind of loops in here, these loops here that are not fully complementary, then you're going to suppress your transcription or the translation of that messenger RNA, but it's not going to degrade it. You're just going to slow down the ability for it to make proteins. Now, all cellular messages are going to be degraded at some point. They're going to be degraded by ribonucleases. The half-life of a message is about 3 hours in eukaryotes, and it's minutes in a prokaryote. Now this is a half-life, so of course some are very short and some are going to be very long. And so this is just in general, they're about 3 hours. Now in the lower eukaryotes, to control messages and to degrade them, there are some approaches they take. They have a short poly A tail. They remove the 5' cap, and so they're going to degrade in a 5' to 3' direction. In eukaryotes, we can target our messages by using 3' to 5' nuclease pathways. And there's also an exosome, which is 3' to 5' exoribonuclease that can degrade our messages as well. Now, I just want to talk a little bit about our RNA-dependent synthesis of RNA and DNA. And so typically, we're going to go from DNA to RNA to, RNA to proteins. And in the case of this, we are going from RNA to DNA. And this is by reverse transcriptase. Now, reverse transcriptase is an enzyme that uses RNA rather than DNA as a template. And we saw this in telomerase. Telomerase does have a reverse transcriptase portion to it. And so you can synthesize DNA using this enzyme from an RNA transcript. There are some RNA viruses that are going to use this strategy to make DNA, such as HIV. And then you're going to, HIV will incorporate their DNA into our DNA. So retroviruses are the viruses that have reverse transcriptase. Now you have your Rous sarcoma virus, which has this SRC gene, sarcoma gene, that's where it gets its name, and this is a tyrosine kinase. And so this has an impact on cell division. And it also is going to interact with cell-to-cell interactions, intracellular communications. This is a proto-oncogene. And it's when associated with a virus, it can lead to unregulated cell division because the virus will express it at high levels. So SRC is a normal gene. When it's in a retrovirus, it is flanked by the long terminal repeats. And these LTRs are strong promoters and you get an increase of the sarcoma virus, the Rous gene, which can lead to cancer. So the LTRs of our retroviruses are very strong promoters for our RNA polymerases. They basically take them over and have them transcribe their virus portions over host proteins. Now, a famous retrovirus, of course, is going to be HIV. HIV is going to cause chronic infection in humans that can lead to AIDS, acquired immune deficiency syndrome. It is going to target and infect your immune cells, the CD4 T cells, and it also has these long terminal repeats. And so it's going to be able to come in as a RNA to DNA through that reverse transcription. And then it incorporates in host genome. And so again, you're going to have the host genome, you'll have the LTR, you'll have HIV, LTR, and then the host genome, flanking on either side. So it becomes part of our DNA, and then those long terminal repeats are going to attract our RNA polymerases to make more of the virus.